Hi, my name is Eduardo and I'm a student in the Machine Learning Pattern Recognition course in Unicamp. Here I'm going to briefly explain the work I've done to tackle Kaggle's 2018 Data Science Bowl competition. The competition consists in segmenting nuclei in medical images. It is a supervised distance segmentation problem, where the images and binary masks are provided. Besides classification, image problems are usually one of three kinds. Object detections, which consist in classifying objects in image along with their bounding boxes of their locations. Image segmentation, on the other hand, makes pixel-wide predictions outputting masks to each class. Similarly, in instance segmentation, the output is a mask, but each instance is individually identified. Therefore, our project requires to identify each nucleus in a medical image, similarly to the picture on the right-hand side. The solution proposed is based on the UNet model. This implementation consists of a convolutional network, which is regarded as an encoder. As we code out the net, the feature maps shrink in size but increase in number. Then, by using transposed convolutions, we reverse the encoding doings by upsampling the feature maps to the original size. Skip connections from encoder to decoder are used to improve the upsampling reconstruction. This model is not used for instance segmentation. It outputs an image segmentation instead. Therefore, post-processing is required for instantiation. Some post-processing are Randall-Walker algorithm and the watershed algorithm. One of the main problems of the UNet is that it fails to predict when nuclei are touching. As we can see from the image in the left, there are many cells that are touching each other. Then, even if the predictions are good, the post-processing algorithm will not be able to identify every single cell in that block. In our implementation, morphological operators were used to obtain a shrunk version of the masks and the touching borders. Then, by feeding multi-channel labels to the UNet, the model is able to learn not only the masks, but also the borders. Now, we have good markers for the watershed algorithm. Here we have the results of some challenging predictions. The problem arrives when there are many cells tied together in the same region, like in the highlighted areas. There are many touching nuclei that were correctly segmented. However, in red, we can see there are some areas that the watershed algorithm failed to predict correctly. This is because even though we implemented UNet to predict the borders, some of the borders predictions are very bad. This is a byproduct of the high data imbalance that exists when we try to predict the borders. Since border pictures are much less frequent than background or nuclei, the network is not able to learn well. This problem can be overcome by using specific loss functions or synthesizing images with cluttered cell blocks as additional training data. For a full read, please refer to the PDF document. The code is also available on GitHub.